The cell cycle is an important process that all cells undergo. It is comprised of three stages, which are interphase, mitosis, and cytokinesis. Interphase is as well comprised of three stages, which are the G1 phase, the S phase, and the G2 phase. During the G1 phase, the decision to begin cell division occurs. The organelles begin preparing to duplicate themselves for when mitosis begins. In order for any of these processes to begin, the cell must first get the OK from the G1 checkpoint, which is located near the end of the G1 phase. During the S phase, the replication of DNA occurs. This is essential in order for mitosis to occur. During the G2 phase, all of the organelles are finished duplicating, all of the DNA is replicated, and the cell has grown to the size necessary for cell division. When the cell has completed interphase, it enters the M phase, also known as mitosis. Mitosis is essential to the process of growth and repair of multicellular organisms. It is a process by which organisms reproduce asexually. Mitosis consists of four stages that each have crucial roles in the cell cycle. During prophase, the chromatin condenses into discrete chromosomes. The nucleoli disappear, and the duplicated chromosomes appear as two identical sister chromatids. The mitotic spindle begins to form, and the centrosomes move away from each other. During metaphase, the centrosomes are now at opposite poles. The chromosomes line up at the middle of the cell, which is also known as the metaphase plate. For each chromosome, the kinetochores are attached to kinetochore microtubules, which is useful for anaphase. During anaphase, the centromeres are separated. This allows the sister chromatids of each pair to separate, making each one a chromosome. Spindle microtubules shorten, causing the chromosomes to move towards opposite poles. During telophase, two daughter nucleoli form in the cell. Nuclear envelopes arise and the nucleoli reappear. The chromosomes become less condensed, and any remaining spindle microtubules are depolymerized. Now, cytokinesis, the third component of the cell cycle, can take place. Cytokinesis is a division of the cytoplasm. For animal cells, a cleavage furrow is formed, which pinches the cell in two. At the end of this entire process, two daughter cells are produced. They are diploid cells, meaning they have the same number of chromosomes as the parent. They are also exactly similar to the parent cell.
Meiosis, like mitosis, is preceded by the duplication of chromosomes. However, this single duplication is followed not by one, but by two consecutive cell divisions called meiosis I and meiosis II. These two divisions result in four daughter cells, each with only half as many chromosomes as the parent cell. Meiosis is important because it allows for genetic coupling of non-identical DNA, resulting in an offspring which has genetic material of two separate individuals. This sharing of material allows for genetic diversity within a population. During prophase I, chromosomes begin to condense and homologous chromosomes pair up. These paired homologs then become physically connected to each other along their lengths by protein structures. This state is called synapsis. During this time, homologous chromosomes exchange corresponding segments of DNA through a process called crossing over. Also, centrosome movement, spindle formation, and nuclear envelope breakdown occurs as in mitosis. During metaphase I, pairs of homologous chromosomes arrange at the metaphase plate. Both chromatids of each homolog are attached to the kinetochore microtubules from one of the poles. During anaphase I, the homologs move towards opposite poles, guided by the spindle apparatus. Cohesion persists at the centromere, causing sister chromatids to move as a unit toward the same pole. During telophase I, two daughter nuclei form in a cell, and each half of the cell has a complete haploid set of duplicated chromosomes. Each chromosome is composed of two sister chromatids. During cytokinesis, the division of the cytoplasm occurs, forming two haploid daughter cells. In animal cells, a cleavage furrow forms, while in plant cells, a cell plate forms. During prophase II, a spindle apparatus forms. Chromosomes are each still composed of two chromatids associated at the centromere. During metaphase II, the chromosomes position themselves at the metaphase plate. The kinetochores of sister chromatids are attached to microtubules extending from opposite poles. During anaphase II, the proteins holding the sister chromatids together at the centromeres are broken down, allowing the chromatids to separate. The chromatids move toward opposite poles as individual chromosomes. During telophase II, nuclei form and the chromosomes begin condensing. During cytokinesis, the cytoplasm divides, forming four daughter cells that are genetically different from one another and from the parent cell. The difference between meiosis I and meiosis II is that meiosis I includes crossing over and synapsis and separates homologous chromosomes, while meiosis II separates sister chromatids.
The differences between mitosis and meiosis are: mitosis has one division, resulting in two cells that have the same number of chromosomes as the parent cell, making them exactly alike. Meiosis has two divisions, resulting in four cells that have half the number of chromosomes as the parent cells, and each one is unique in their own way.